Hello to all, and thank you for joining me in this presentation. Today I'm going to talk about temporal consistency on aggressive behavior in an ectatominae ant, which is part of my doctorate thesis. When we talk about behavior, it is important to remember that individuals vary between them in the way that they behave, but these behavioral differences between individuals can remain consistent over time in different contexts, which is the concept in which we define animal personality. It is precisely this definition of personality one great limitation of behavioral studies, because to discuss personality, one needs to discuss temporal consistency, and for that, we need methods that have repetition in time. We neglect the importance of repetition when working with behavior, not only in personality studies, especially because behavior can be displayed in a continuous range and be influenced by many environmental and physiological factors, and with more repetition can be an efficient tool to counterbalancing that. A lot of studies rely on one or two temporal repetitions only. But with only that, can we really discuss repeatability or even consistency of behavior? Fewer repetitions sometimes can bias interpretations of behavior patterns. More repetition, even if possibly including more individual behavior variation, can provide a better understanding on individual behavior patterns. Aggressive behaviors are shown to be one of major components on personality traits, and many studies have been shown to correlate these traits with many others, such as sociability, boldness, proactivity, many others. This trait is especially important when working with social insects, more so with ants, where aggressiveness can play a major role on nest defense and search for resources. Social insects are also an interesting model when working with personality for singular traits such as genetic similarity or colonial level personality. On this study, we work with an ectatominae ant called Gnaptogenes striatula, ants that occur throughout South and Central America that, like most species of this genus, are generalist predators. Considering all these factors, our study aimed to answer the questions: do individuals of Gnaptogenes striatula show temporal consistency of aggressive behavior? For that, nest collections were carried out in University of São Paulo, Brazil, in October 2020. In total, 2 nests were collected, and from them, 6 subcolonies were assembled, each one with a start combination of 250 workers, 20 larvae, 20 pupae, and 20 eggs. Only foragers underwent the regression tests and were selected through behavior observation. They were individually marked with a dot of ink in the abdomen and the thorax, allowing individual discrimination. A minimum of 5 individuals were chosen per nest to undergo the aggression tests. Aggression tests were carried out on a petri dish of 9 cm diameter with a peripheral area marked in pencil, 1 cm away from the rim, with a clean filter paper floor that was changed for each trial. The ant was introduced in an acclimation tube for 60 seconds, while a dead stimulus was placed diametrically opposed at the center of the arena. Then the tube was removed, and the behaviors were recorded for 300 seconds. A circle of 1.5 cm diameter was also delimited around the stimulus to calculate the stimulus peripheral zone. The heterospecific stimulus chosen was a dead worker from Solenopsis, from a colony that was also collected at University of São Paulo. Individuals were frozen right after field collection, and for the trials, individuals that would be presented as a stimulus were defrosted 30 minutes before the test. This test was repeated 5 times throughout 3 weeks for each individual. All encounters were video recorded. After data collection, videos were analyzed on board software, and aggressive behaviors were categorized for duration and frequency and coded for their aggressive scale. And after that, an aggression index was calculated for each repetition for each individual. We also calculated the latency to first intonation and to enter stimulus periphery. And with the Agile Vision software, we calculated the time spent in different arena zones and the distance moved. Out of 154 individuals that were initially marked and underwent the first aggression tests, Only 38 individuals reached the 5 repetition criterion, mainly due to death or loss of individual identification. Analyses were carried out in the R software with the RPTR package to calculate individual repetition throughout trials. To understand how repeatability changes with increasing repetition, we also carried out the repetition analysis for 3, 4, and 5 repetitions for the same individuals. Repeatability was found for several parameter tests, starting with our variables with 5 repetitions that were latency to intonation, latency to enter stimulus periphery, total time in intonation, total time in interaction, total distance moved, time spent on arena periphery, time spent on stimulus periphery, and aggressive index. Looking closely to the aggressive index, we can see that though repeatable throughout time, aggression was low for the great majority of the encounters, mainly because the great majority of the interactions behaviors were intonation, that represented an aggression score of 1, or no interaction at all, with aggression score of 0. That's also the reason that we use total time and interaction as a variable instead of each behavior separately, because interactions were short and mainly based on intonation, when it occurred. Latency to enter stimulus periphery was also repeatable, with no effect of trial number on this variable. Here, the mean latency for each trial represented with a black dot. This proposes the idea that no learning or sensibilization effect affected the time it took for individuals to approach the stimulus throughout time. Here, we demonstrate a graphic representation of what we refer as individuals: low, blue dots with blue tendency lines; median, pink dots with pink tendency lines; and high repeatability, gray dots with gray tendency lines. To understand how increasing repetition can influence on repeatability parameters, we analyzed the same aggression variables, but with the data of also 3 and 4 repetitions to compare with our data of 5 repetitions. We found that for the p-value, there was not much of an influence of the increasing repetitions, for all the values remain statistically repeatable, with only the aggression index lowering the p-value with increasing repetitions, but still all remaining below 0.05. For the R values, we have a similar pattern, where increasing repetitions didn't impact much of the variable value. We see a slight increase of R value for total intonation time, but for the rest, it remained pretty similar. The variable that have shown to be the most affected by increasing repeatability was the confidence interval. Here, we see plotted the maximum and minimum of confidence interval values for each variable for each set of repetition. For all the variables, we see a pattern that increasing repetitions lower the range of confidence intervals, resulting in a more narrow confidence interval with 5 repetitions, which in turn can represent that our study behavior variable is closer to represent the real behaviors displayed by the individual. So, going back to our first goal of the study, we have shown that individuals of Gnaptogenesis striatula show temporal consistency of aggressive and many other behaviors, although individuals show low interspecific aggression, with intonation being the most common of the interactions behavior. Other studies have found the phenomenon of low aggression for this species as well, although being discussed in a different context, such as dominance hierarchy. But still, maybe we can discuss the idea that low aggression is a throughout species trait for Gnaptogenesis striatula. Like we said previously, repetition is based for discussing animal personality, and although it imposes methodological limitation, as we've seen, out of 154 individuals, only 38 reached the end of 5 repetitions, and we know that many species have more restricted limitations regarding sample size or lifespan, but in spite of that, we've shown that increasing repetition can have great effect on behavioral data, in spite of not having great effect on the R, or P can narrow the confidence interval of this repetitions analysis, displaying more clearly behavior patterns. This, however, goes in contrast with other studies, for example, that study short and long-term repeatability of aggression and other behavior traits in social spiders. They show that there was a reduction on repeatability of personality traits over the long term relative to short term. But in spite of that, Long-term personality consistency can be found in many other studies for a variety of species, so maybe we can discuss how methods can be more adapted for the lifespan or behavior displayed by the species and how we could accurately assess them. With these results, we can also discuss that Gnathogenesis tiratula is a possible good study species for animal personality, since it can display temporal behavior repeatability for more than one subset of behaviors. We require further testing for other behavior traits to see if this repeatability is also found, and investigate further the possibility of correlation between behavioral axes. That's it for today. Thank you all for watching this presentation. Please feel free to contact me if you have any questions. Bye!